So thank you again, Catherine, for the fantastic conversation. And our second guest we already have is Dr. Thomas Merritt. Dr. Merritt is a Drosophila scientist. I'll let you actually start the conversation with by introducing yourself.
Who I am?
Yeah.
So my name is Thomas Merritt.
Yeah.
I'm a faculty member and professor at Laurentian University in Northern Ontario, where we work on the genetics of stress using fruit flies and a various suite of stressors.
Awesome. So, and you're also a Canada Research Chair.
I'm a past Canada Research Chair.
Oh, really? Okay, cool. But you're in the Department of Chemistry and Biochemistry.
Past member of the
Oh,
department.
really? Oh, okay.
So I'm
Interesting. I didn't know the system.
So we're now the School of Natural Sciences.
Oh, I see. So that's the old name, and now, okay, yeah. So Thomas was the guest, well, you were the guest for our 5th episode of our podcast titled Happy Accidents in the Lab. So, and that was published in September 2023. So if you haven't listened to our podcast, Genetics in Your World, I really recommend the Twitch streamers to really plug onto it because it was such a good conversation. You shared the story of regulation at the Drosophila's Malic enzyme highlights the complexity of transvection and its sensitivity to genetic background, right?
Yeah.
So that was published in the journal Genetics in February 2023. So can you tell us more about the project that you were publishing, like that you have published there, and is there any updates on the project?
Sure, yeah. So I'll give you a little context. So
Sure,
the paper's a really nice paper. It was an editor's choice in genetics, I think, a year ago.
It was a
right
featured
now.
journal, an article.
And I think it was featured because it's a phenomenon that was described in the 1950s. So it's called transvection, and that doesn't necessarily mean anything to most people, but it was one of the earliest forms of epigenetic regulation. And so now when we talk about epigenetics, above genetics, we're almost always talking about chemical modification of histones, chemical modification of DNA. But that term predates our ability to do things like identify chemical modification. So it basically comes down to epigenetics, comes down to things that our typical models of genetic regulation don't explain. And so transvection was this early thing that was a head scratcher. And there was a lot of just classic genetics that was done looking at this phenomenon. What we now know happens is, we studied fruit flies. It's really common in fruit flies. It probably happens in many other organisms as well. Fruit flies, like us, are diploids. They've got 2 copies of every chromosome. And largely because we misinterpret Mendel, we always think of these chromosomes as independent, right? And they're inherited independently, but they don't function independently. And so there's this sort of disconnect between when we teach Mendelian genetics and when we think about Mendelian genetics. Transvection is a really great, clear, distinct case where the chromosomes are not independent and they actually physically interact. So in fruit flies, it's really pronounced because in flies, those chromosomes line up really cleanly. And so there's this physical interaction between the pairs of chromosomes. What we now know is that physical interaction leads to a sharing of regulatory elements. So if you think of a gene, right, and just even to like the classic, one gene, one protein, we know it's a simplification, but you've got this strand of DNA that's a code for the protein. That DNA is the gene, the code is in the DNA. The biology is over here in the protein, so we have to get between those two. So you have to turn on the gene, express it in the right tissues. In the typical model, you've got a regulatory element, you've got a gene, and we're going to turn the gene on, up, off, whatever it is. In transvection, what happens is the regulatory elements here on this chromosome actually talk to the other chromosome.
On the other chromosome, not even like far away that we learn that enhancers can be several hundred base pairs upstream. It's not even on the same chromosome. Hundreds of thousands, yes. Yeah, so they're not even on the same chromosome.
That's right.
And so it's so mind-blowing. And I believe there would be so many, I don't understand this because they're not even on the same chromosome, but you have to have,
And so one of the things, though, I think is if you take a step back and you haven't been told how gene regulation works, it's not that surprising. Like, okay, so you've got 2 switches, they're not independent, the 2 switches interact. But we always teach the fact that the 2 switches, the switch on this chromosome, the switch on this chromosome, are independent.
So often we teach a very simplified version.
That's right. Yeah. And so the paper that we had, the student Teresa Gisnichuk really clearly showed a case of transvection, but what she showed was that the amount of transvection, so what happens in transvection is in our case, the system that we've set up, there's a dead gene, so a knockout gene, and there's a functional gene. And this interaction upregulates the remaining functional copy. And what she showed was that across individuals, and we looked at something like 150 different lines of flies, the amount of upregulation, so essentially misregulation, can vary tenfold. And so we think we have the answer looking at this line, and then we look at a different line, and the response is tenfold higher. Or we look at another line, and the response is not there at all. So you go from anywhere from absolutely no transvection to sort of our typical levels of transvection, which upregulate to sort of a normal level of gene expression, to higher than that. And so there's this huge variation, and we went back and tried to find the genes that were involved with that. We had more luck saying that there's an enormous amount of variation than we had in finding the genes that are actually driving that.
The one gene driving the transvection.
That's right.
But the genetic backgrounds really matter.
Enormously
matter.
And that's another thing that in genetics, like when we work in model organisms, we often discount that. And so we'll work on a line of flies. And what Teresa really clearly showed was that the line of flies you're working on really matters. And if you want to understand the biology, you can't work on a line of flies. And maybe you don't always work with 120 lines of flies, but you need to factor in the same ways that, like, if you're going to understand biology, you have to work in males and females. You can't just work in males and just work in females and understand the biology.
It's just showing one side of the picture,
And so in the same way, when you work with lines of flies, you should be working with multiple lines of flies. And the bigger picture is no matter what you work on. So if you're working in humans, if you're working in mice, if you're working in any system, you should be working with both sexes, if there's sexes in the system, and you should work with multiple lines of whatever the organism, multiple lines of mice, C. elegans, whatever that is.
Let's get some questions from the stream. There are really good questions. So first, kind of like backing up a little bit, why do you work with flies, and are they like us? Are they like me?
So yes, and there are lots of reasons. So flies are actually very similar biochemically, metabolically. So traditionally we worked on the genetics of metabolism and metabolic responses to stress, and this gene regulation story actually came out of a failed experiment. And maybe we can talk about that a little bit, how to use a failed experiment to explore something novel. But we were working on flies because a lot of fly metabolism really mirrors what we see in humans. And we can do things in flies that either we wouldn't do or we can't do in humans. So actually one of my students who's here presenting part of her master's work, she's getting ready to defend, so she's putting together her slides. And I was like, okay, Allie, how many flies have you've been working with? Because sometimes this is a really good contest. And she's like, okay, well, I worked with about 12,000 flies from undergrad. She did the math and she came back, she goes, we worked with 43,000 flies in this experiment. Right? And so individual variation really matters. So one of the reasons we work on flies, they're small, they're cheap, they're easy to work with, and all of that translates into sample sizes that are absolutely ridiculous. So if we were interested in studying this kind of regulation in humans, we would be experimentally manipulating 43,000 people. So on the one hand, we wouldn't experimentally manipulate people, and on the other hand, that's just too expensive. And even in things like mice, working with tens of thousands of mice becomes prohibitively expensive. Your last guest, there are times where people who work on yeast, C. elegans, bacteria, I have model species envy because they're working with huge numbers of bacteria, yeast, C. elegans. So bacteria, fruit flies and humans are actually relatively similar, they're relatively similar. A lot of the reasons we work on flies are because they're easy to work on and we can ask questions in a fly model that we don't get an opportunity to work to ask in other systems.
And the genetics is so sophisticated. We have so many tools. There are only 4 chromosomes, opposed to like other organisms, they have like few dozens of chromosomes in their organism, even though they are a simple system. So I think those are really, well, I'm a fly person, by the way, just a disclaimer, so I'm like very biased onto the flies.
They're a great system.
They are a great system. So they are, they're a great system to study fundamental biology.
That's right.
That can be applied to humans. So what does it mean classic genetics. So you mentioned about Mendelian genetics, and so there was this comment, wait, Mendel wasn't right? So how would you respond to that?
Okay, so it depends on how far you want to follow down that rabbit hole. Mendel probably lied. It's not mathematically impossible for Mendel to have come up with his results, but it's really unlikely that Mendel actually came up with his results. So he gets these really beautiful ratios of 4 to 3 to 1. The numbers are so close to what you would, if you were to just to create an example.
In an ideal, very ideal situation.
So that's problematic. There's many layers to this. The idea of Mendelian inheritance. So alleles at a particular locus are inherited independently. Absolutely fundamentally true and incredibly important. The idea that genetic variation at different loci, so different genes, is inherited independently. True, absolutely fundamentally important. The data that he presented probably made it up. I mean, it's based on experiments that he did, but he probably sort of rounded some stuff.
I mean, Mendel was super lucky to choose those traits that really followed the simple genetics, but in real life, it's not always the case, I guess.
It also, so peas have 7 chromosomes. Given the techniques that Mendel was using, to see the independence of inheritance, those traits had to be on separate chromosomes. He just happened to pick 7 traits that were on 7 different chromosomes. Highly unlikely, and it's just, the 8th trait that was on the same chromosome as the first trait, he buried the notebook somewhere in the backyard. So, without going into too many of the details, the fundamentals of Mendelian inheritance are absolutely true, and they really are the foundation of modern genetics. The numbers are really too good to be true, and many, many different levels. The other part of that, though, is Mendelian inheritance is 100% true. The way that we think of Mendelian biology is not. Any given trait that we can think of, the alleles behind that trait are inherited in a Mendelian fashion. They don't function through Mendelian inheritance. So, like a Mendelian trait, so you mentioned the fact that he had these great things. So yellow peas, green peas, wrinkled peas. So Mendel's peas were wrinkled, fine. The biology behind that is more complicated than that pattern of inheritance. Most traits are in fact controlled by multiple genes. And so eye color is the example that I normally use. And so we sometimes think about, blue eyes and brown eyes. And as a relatively simple case there, we think we know the gene that's involved. We even think we know the mutation that happened about 5,500 years ago. And that's actually sort of interesting, that blue eyes are not old. Blue eyes are probably less than 6,000 years old.
Evolutionarily.
Evolutionarily.
Yeah.
But yet, it's not that my eyes are 5,000 years old. Thank you for correcting me. But that mutation is a relatively recent mutation. But if you look at my beautiful blue eyes, and you look at my kids' beautiful blue eyes, they're slightly different. And so my kids' eyes are slightly grayer than mine. And there are at least 14 or 15 other genes that interact. And so the genetic variation across all of these 15, 16, 14 different genes gives us that final trait. We sometimes miss that when we teach classical genetics. Mendelian genetics explains inheritance. It doesn't explain functional biology. Functional biology is complex traits. And it's multiple genes coming together, interacting with the environment, and interacting with the flip of the coin.
Absolutely.
And that last one is the hard one. So there's a certain amount of just random chance in the way that genes work, and we sometimes have a hard time appreciating that.
Yeah. Thank you for that insight. Like often, I teach
intro.
Eye joke? Sorry?
Is that insight? Is that an eye joke?
It's no.
Sure.
No. So I teach intro biology in my place, and so we talk about Mendelian genetics, and we also talk about non-Mendelian genetics, that like the pleiotrophy that you talked about, like multiple genes having different phenotypes, or multiple genes contributing to one phenotype, or multiple phenotypes that are elicited from one gene.
Absolutely.
All those kind of like more complicated things in genetics, like that's what happens in our bodies. That's how we get this much variety of life, and phenomena. But I think it's kind of like really cool to think that like the research that's happening in your lab is really tackling those like precise questions on like how the genetics is actually happening
Sure.
in these in these different backgrounds.
So just before we leave that point,
sure.
like addressing that reality is super important, because when you teach people like an oversimplified Mendelian genetics, and then they even look at something as simple as eye color, they're like, well, obviously it doesn't work that way. And so it undermines our appreciation for science unless you say, look, here's a really simple model, but biology is complicated. So we can start with a really simple view even of eye color, but then we tie in the reality of complexity, and like, okay, why haven't you cured cancer? Because it's really complicated.
And the backgrounds are so different. I think you really touch on a super important issue that we can talk forever, but the fact about even skin color, the variations in our genetic information in the nucleotides don't really translate into one phenotype. So some people there are these stereotypic thinkings on genetic variations and how that comes from like few genes that are different in us. Like, I mean, humans, we all have like less than 0.01% difference in our genetic variances, but we have this much of differences in our phenotypes. So it's really hard to like grasp on that, thinking only that like, if you only follow the classical Mendelian inheritance. So I think the introduction of this variations, like on top of the genetic variations, that's really coming and it makes the whole big picture and more colorful, I guess.
That's right.
Yeah.
Absolutely. That idea of how similar we are. Our genomes are really big, right? So we've got something like 3 billion units of information in our genome. Well, even with that low level of variation between any two of us, there are 6 million differences. So if we were to sequence my genome and your genome and compare them, there's 6 million differences there.
Yeah, still.
So, some of them are going to be really, really simple things like, why are you female and I'm male? So why do you have brown eyes and why do I have blue eyes? But there are going to be other things like, why do you phrase sentences in a certain way, right? So that's going to be how you grew up, the languages you spoke, but also just sort of fundamental parts of your personality that really probably are genetic.
Right.
And that's tucked into those 6 million differences.
But it's not like a single gene that is turning on and off.
And it's not even a single switch. It's a switch that's modified. It's 100 switches coming together, modified by the environment and your past history.
And it's not an on-off switch. It's like a more
it's a dimmer.
lever.
It's definitely
absolutely.
a dimmer.
Yes. So there is a comment that says like, "Yay, I'm new," which is a referencing to the eye color, blue eyes.
There you go. Yeah, that's right.
Yeah. So we're more newer humankind, I guess. So what is a line of a fly?
Oh,
do they
sorry.
dance?
Yeah,
do
that's
they dance?
jargon. Oh,
So this is this is a really great point. Thank you for the comment. And what is a line of a fly? Do they dance in a line?
Yes, they do. So it's actually based on line dancing. So it's a recent term, came out of country music in the late 80s, and the flies line up on the dance floor. It's not true.
And you can see under the microscope.
And you can. Actually, there are mutations where the flies do line up.
The fruitless?
Fruitless is what I worked on as a grad student.
Cool.
Okay, so a line of flies is essentially a family of flies. And so we work on multiple lines of flies. Different lines of flies have different relatedness within a line, but essentially it's a stock. So you could think of like a
interbred,
but they're not always interbred. That's why I'm sort of hedging.
Okay.
No,
Right.
that's why I'm sort of hedging around it. So if you think of like different, what's a good way to do it? So they're all the same species. In my lab anyway, we've got multiple lines of Drosophila and melanogaster. A line is a group of flies. Sometimes they're genetically related. In some cases, they're what we call isogenic, and every fly in a vial is genetically identical. Other times they're not. And so you mentioned compost and C. elegans. We actually work with flies from the composter in my backyard. So fruit flies, Drosophila melanogaster, they're an African species, but they spread globally. One of the ways that we look at these populations of flies is we take individual lines, which are like a snapshot of a population. And so in some cases, those lines are genetically diverse, and it's just it's a set that may have been anywhere from 2 to 100 flies in a vial that we've maintained. They're flies, they're sort of classic lines of flies, Cantoness, Organar, that you've probably worked with. Some of them have been isolated since the early 1900s.
Yeah.
And they're more machine than fly. They're a really messed up organism. They're sort of the HeLa cells of the fly world. So a line is some kind of representation of flies in general. Oftentimes they're a snapshot of genetic diversity. Sometimes they themselves are diverse. So we have diverse lines in the lab, and we have lines in the lab that are not diverse.
Cool. Yeah. Thank you for that clarification. Can we talk a little bit more about your failed experiment? And I imagine a fire, you know?
Yeah. So, one of the things, and so you and I talked about this a little bit. So I ended up writing a piece on the importance of celebrating scientific failures. And so in this case, I did an experiment in somewhere around 2003 where we had genetically engineered flies to create a difference in enzyme activity. And when we did the genetic engineering and we did all the preliminary experiments, we had normal flies and we had knockout flies, and they worked beautifully. And we crossed the two of them together, and it simply, we're expecting an additive. So if you have 0, you have 100, the heterozygotes that cross should all be 50. And they were more like 80, 85% instead of 50. And so that was the failure, because we
totally unexpected.
Totally unexpected, and we thought we wanted to have 50 and 100. What it made us do was go back and say, okay, well, why did we think it was 50? And is there an alternative explanation? So did we just screw something up, or were we making an assumption? And the assumption we were making was that the two chromosomes were independent.
Yeah.
And luckily, my advisor, Walt Deans, is a really great Drosophilist and had been doing this long enough that he said, hey, I bet that's transvection. And that's this interaction between the chromosomes. So the experiment failed to generate what we wanted, but it made us question the assumptions, and in doing so, went back and said, oh, we're assuming the chromosomes are independent. What if they're not? So 20-something years later, we're still studying that. And so Teresa's paper last year, the really important contribution that she made was to say, not only are the chromosomes not independent, they're physically interacting, but those physical interactions are modified by genes that are spread across the rest of the genome. Like many, many genes across every single chromosome that are modifying those interactions. So we assumed they were independent. We were wrong. That failed experiment made us question that. We assumed that the way they interacted would be constant across different individuals, different lines of flies. We were wrong, and when we looked at it closely, it ends up we were really wrong, and there's a huge amount of variation.
How much of the proximity, like they have to come close to actually have that physical interaction?
That's super cool. So the idea is the chromosomes physically line up. And we know this because in fruit flies, well, in essentially all maybe organisms, you have variation where chromosomes are flipped. We call them inversions, right? So in fruit flies, the inversions are relatively common. And if you have an inverted chromosome and a typical chromosome, they never interact. And so that transvection goes away. And so we actually showed that in a paper in maybe 2010, 2011, that when you invert the chromosome, so that physical space is changed. So your question though is like how close? It ends up, we don't know. And we don't know in really interesting ways. So there are inversions that will do away with transvection, and there are other inversions that when we look at the landmarks, the places of the inversion, we would predict that transvection would go away, and it doesn't.
Those breakpoints are not important then.
They're just
it
not.
It would be an easy target to like go and investigate first, but
that's right.
That doesn't work.
Yeah, and so there are definitely some rules as to governing these physical interactions, but what the rules are, we're not really sure. And so that's sort of a long-term study, and people have been working on how do these breakpoints work? How do these inversions work since the 1950s?
Is there other like proteins that are interacting with the two chromosomes, like the kinetochore?
Well, there are probably some large-scale things, but there are also some small-scale things.
Oh, I see.
And so what Teresa was trying to do with her work is another student in the lab had shown that when we create different lesions in the chromosome, different holes in the chromosome, we get different amounts of transvection. And so we were predicting that what we were doing was deleting different regulatory elements. And so if the regulatory element is missing, then a regulatory protein can't bind to it. And so we were thinking with the mapping that Teresa did, that we'd be able to identify across the genome proteins that were then binding at the site of transvection and modifying that transvection. it might be more spatial, and so it's not the presence or absence of a binding site, but it's actually twisting the chromosome itself, and that physical distortion is leading to the transvection.
So this is not recombination. It doesn't really like switch the genes from different chromosomes, but one chromosome is influencing the other chromosome.
You're entirely correct, and that's actually, so that question is a
Yeah.
very well-informed question, because it shows that, and this is, when I've talked about this work, people are like, well, isn't that just recombination? So it's happening in the soma, it's happening in the adults, and it's not permanent. And so if we take those flies that have transvection and then we cross them to another generation of flies, you'll recover wild-type expression in one case and knockouts in the other case. So the alleles are still moving independently, they're just not functioning independently.
Interesting. That is so interesting. Yeah, I have this really good comment. I wish this guy was my professor. Yay. And what is the coolest thing you have ever found? So probably related to, but it could be something that happened like in a totally different experience.
Yeah, actually, so that's honestly the coolest thing I've ever found is transvection, because it made us really made us question what are the assumptions that we're dealing with. And we were assuming that chromosomes are independent because that's the way we teach chromosomes, and chromosomes are not independent. And I still struggle to pitch that to people. It's not, if you work in the field of genetics, it's going to be a really hard thing to appreciate. We've had some really great work recently. Ali Sutherland Hutchins is talking about it here with the amount of variation that we find in like stress response, how fundamentally different it is between males and females. And that's also been a hard thing to grasp, that the biology of males and females is really distinct. And historically in fruit flies, we typically have only worked in males. And even when I was a postdoc, there was this general pitch that you just work on male samples, there's less variation, and females are just hypervariable males. And so if you understand what's going on here in males, it's the same thing in females, it's just a little bit more variable. And the more that we actually look at it, like, yeah, it's not.
It's not, right?
It's
The
really
sexual
not.
dimorphism is like so huge. Yeah.
And so work that Ali's presented in a poster this afternoon, I think that's been almost as much fun as the transvection, because it's really driving home the idea that the two sexes share so much of their genetics, but the biology is so incredibly distinct, and really pushing that you have to be working in both sexes.
Absolutely. So is there a difference in the transvection in the same line versus like female versus male?
So we actually understand less of that than we do with some of the other phenotypes. I think that there is, we haven't studied as closely as we have some of them.
I see, but that would be really interesting because in flies, males don't go under recombination.
That's right.
So I don't know, like, but it might be related.
Yeah, it would be a really interesting thing to look at. That's absolutely true.
Yeah, that would be really fun. Cool. So I guess as a wrap up, like, can you share a little bit about your future goals or your other, like, wrap up anything that you wanted to add on to?
So what are we doing now? Like, where are things
what
going
are you
now?
doing now?
So a lot of the transvection work really drove home the line dependence of the biology we're working on. So right now, most of the lab is working on nickel stress. So we work on stress biology. The stressor that we've been focusing on for the last couple of years is environmental stress using nickel as a toxin. So Sudbury, Ontario, where my university is, at one point was the nickel mining capital of the world. So nickel contamination is sort of a local topic that people are interested in. So we've been doing a lot of work looking at individual line variations of this idea of these different snapshots of variation, and really finding like enormous differences in resistance. And one of the experiments that Ali's going to talk about this afternoon that was really fun was we've got sensitive flies and we've got resistant flies, and we're like, well, what happens when you cross them? Are they heterozygous? And what we expected was everything to sort of move back to the middle. So if you're two extremes, you're really resistant, you're really sensitive, you cross them, you think everything's going to be in here. At least at first pass, what happened was she crossed the resistant to resistant, and she created flies that you can't kill. Like, these things are just,
super resistant.
They're metal impervious. And we're looking at the results, it's like, do you believe that? It's like, I don't know, we got to do that again. So there's always some new
There's
twist.
the failure again.
Yeah, and so it's like, okay, well,
yeah.
we failed to get these flies.
Yeah.
Like, can you kill the flies? So like, we actually can't get enough nickel in the food. Like, we hit the limit of solubility, and the flies just cruise along happily on this like high nickel food. Like, okay, so we're going to keep doing a lot of work with the sex-specific response. There's another students now working on the microbiome, so the bacteria that live in the fly. And some of the nickel biology that we're studying, we think we're actually studying microbiome biology.
That would be, really cool cross-section between, the interorganismal
That's right,
communication.
so interorganismal communication is super cool.
That would be really cool. I work in gut epithelium,
Oh,
so that would be like super interesting
Yeah.
to talk more about. And there are, specific compartments within the gut that is responsible.
Yeah, well, okay, we should chat.
Yeah,
because
we
that's
should
something
chat. That would be super
Jesse's
fun.
struggling with.
Yeah, totally. So I guess with that, there's, everybody, I guess I can finish with this comment. Everybody's worried about AI, but metal impervious superflies are the real danger out in the
There
world.
we go.
So
We'll end with that, that's awesome.
Thank you so much for this conversation.
Thank you, this has been fantastic.
Yeah, thank you so much.